like to start off by thanking the IMP and the Birnstiel Foundation for honoring me with this PhD award. And I'd also like to thank them for the opportunity to speak about my PhD work today. So today I'm going to tell you about the focus of my PhD, which was how are membrane proteins made, and more specifically, how are multipass membrane proteins made. Now this is a specific subclass of membrane proteins which contain more than one transmembrane domain. And this has been a long-standing question in the field, but we do know a few things from the study of simpler membrane proteins which contain maybe one or two transmembrane domains. The process starts in the cytoplasm when a ribosome is synthesizing a nascent chain that encodes for a membrane protein. When the first hydrophobic element leaves the ribosome exit tunnel, now this can either be a signal peptide or a transmembrane domain, it is recognized by the signal recognition particle, which shields this hydrophobic element until it is then delivered to the endoplasmic reticulum through its cognate receptor, the SRP receptor. Then, through a handoff mechanism, which is not quite understood yet, the transmembrane domain engages Sec61, which acts as a sort of molecular conduit from the cytoplasm to the hydrophobic environment of the lipid bilayer. The transmembrane domain then moves laterally into the lipid bilayer, and that constitutes insertion of a transmembrane domain. Now, we know that the following TMDs are then inserted afterwards in a sequential manner until you finally get insertion of the full transmembrane protein. However, there are two really big remaining questions. The first is, when a transmembrane domain reaches the Sec61 channel, how is the multipass membrane protein orientation determined? In other words, how is its topogenesis actually determined by the insertion machinery? And the second question is, once you've now inserted all these transmembrane domains in a sort of linear fashion, how is it then folded into a three-dimensional structure? Now, as a young, somewhat naive PhD student, I didn't really appreciate the importance of this first question of topogenesis, and I was really focused on this question of folding and assembly. I thought this was really an important question. So the hypothesis has always been that it's analogous to how it works in the cytoplasm, in that you have chaperones which facilitate the folding in the plane of the membrane, but none have ever been identified. So we first took a very focused candidate approach, focusing on this newly identified complex called this ER membrane protein complex. So the ER membrane protein complex was originally identified through a synthetic genetic screen to be associated with things that are known to participate in membrane protein biogenesis. And so we thought this represented a good candidate that might be a chaperone, but what we ended up finding was that EMC is actually acting at a very early step to properly insert and orient the first transmembrane domain of a subset of multipass membrane proteins. And these multipass membrane proteins are ones in which their N-terminus is located in the lumen of the endoplasmic reticulum when they're inserted, and then the C-terminus following that first transmembrane domain is actually in the cytoplasm. And we call these type 1 proteins, and it turns out EMC is completely necessary for their insertion. And without EMC, you disrupt insertion, which it fails altogether, or you get insertion in the opposite orientation. So then after figuring out that EMC was actually acting at this early step, then we were really interested in what happens further downstream, and that is, how is a membrane protein folded? And so we went searching for previously unidentified chaperones in the membrane. And what we found was this novel intramembrane chaperone complex, which we're calling the PAT complex. And it turns out it actually binds to transmembrane domains based on the presence of hydrophilic residues, remains bound until the entire protein is synthesized and has a chance to fold into a three-dimensional structure. So I don't have time to go into all the data today, so I'll just give you a few highlights on why we think this is, which points to why we think this is what's going on. So when first starting, the first thing we needed was a system by which we could interrogate multipass membrane protein biogenesis, and to this we turned to in vitro translation assays. So the way this works is, you essentially have a cell lysate in a test tube which has everything you need to synthesize proteins. You then add your mRNA of choice. In this case, we're looking at the GPCR, the beta-1 adrenergic receptor, which contains 7 transmembrane domains. And then you also spike in a little bit of radioactive methionine so you can visualize this on a gel. And you can see here when we actually translate in vitro this beta-1, we get a nice clear band here. And then if we then take ER, which we've isolated from wild-type human cells, and we add that into the translation reaction, we can get insertion of the membrane protein. And we know that we're getting insertion for a few reasons. The first is we get this glycosylation here. And glycosylation occurs in the lumen of the endoplasmic reticulum, and we know that the glycosylation site of the beta-1 adrenergic receptor is on the N-terminus. Additionally, we can use ligand binding assays to determine that this protein is properly folded. And I should point out here that we actually know that essentially the entire proportion of this glycosylated population here is actually folded. Now, what happens if we take away EMC? If we delete EMC genetically and then isolate ER from human cells and include that in the translation reaction, what we see is a dramatic reduction in this glycosylated population. Now that's important. It tells us two things. First, it tells us that the N-terminus is not quite in the right location for some reason. And it also tells us that beta-1 adrenergic receptor is probably misfolded. And we can actually confirm this if we look now in cells using a live cell assay and determining the stability of the beta-1 when we transfect it into human cells. So the way this assay works is it's actually a ratiometric fluorescent assay. So what we've done is we've tagged the beta-1 with a GFP, and then we have a cytosolic reporter RFP molecule, which is indicative of complete translation. So any reduction in the ratio of GFP to RFP indicates degradation of the beta-1 adrenergic receptor. And so what we can see here, if we do this in cells, we've got wild type cells here in gray, and what this establishes is a nice baseline ratio of beta-1 adrenergic receptor stability. If we then delete EMC, we see a dramatic reduction in beta-1 levels, and so that's indicative of degradation. We can then rescue this completely by acutely re-expressing EMC under a doxycycline promoter. So at this point, EMC is actually looking like a very good candidate for a chaperone. It affects membrane protein biogenesis, both in vitro and in vivo. However, when we dug a little deeper, we could determine that EMC is actually acting at an early step. It's actually acting on the first transmembrane domain of the beta-1 adrenergic receptor. And we can see this here if we just take the first transmembrane domain into our in vitro translation assay, and we see that we still see this glycosylation defect, which we saw the full-length protein. If we now look into the topology in either case, we actually see that this glycosylated portion that we see that is enriched in the EMC knockout ER actually is just an inverted insertion. So it's actually inserted in the wrong orientation. So in this way, EMC is actually important for establishing the orientation of the first transmembrane domain, which, if you remember, is actually a critical step in establishing the topology of the entire multipass membrane protein. Now, just a few more highlights, which I don't have time to get into. We know that EMC inserts the first transmembrane domain. If we use purified EMC, we can actually show that it's sufficient to insert that first transmembrane domain. And we know that it actually impacts multiple GPCRs. We tested about 17 GPCRs, and all of them are impacted by loss of EMC, and their insertion is impaired. And very importantly, if we try to insert, we know that EMC is not actually needed at later steps. So if we force correct insertion of the first transmembrane domain, in the absence of EMC, the second transmembrane domain is inserted just fine. So then the question is, what happens next? And what I mean by that is, what happens when these transmembrane domains are all inserted correctly, but then the protein needs to fold, and which factors are really facilitating that? So for this, we made use of a little trick, which is that you can translate a truncated mRNA, which stop codon has been removed, and stall translation at a very specific point. So in this case, we stalled translation in which 2 transmembrane domains had been inserted to the ER, but it's still attached to the ribosome. Then we took advantage of chemical cross-linking by placing a single cysteine in the first transmembrane domain and simply asked whether or not we could see interactors with this first transmembrane domain. And what we found was this very robust interaction that is roughly 10 kilodaltons in size, which we saw was previously very similar to a cross-link that had been observed by Stephen Hai's group in the early 2000s. And so this protein we're calling PAT10. And then very nicely here, just to show this assay is working quite well, we can actually see a nice cross-link here to Sec61. All right, when we then tried to identify what this PAT10 protein was, it turns out it was quite difficult to identify this very small 10 kilodalton protein directly. So instead we looked for all interacting proteins of this cross-link by solubilizing under native conditions. And what we found is that it was actually part of a complex. And this complex consists of 2 proteins that are essentially in stoichiometric quantities. And we see that is Asterix, which we identified as the small PAT10 protein, and CCDC47, which is a much larger protein. And so then the question is whether or not this is actually acting as a true chaperone complex. Now, there's a handful of experiments that we've done to show that this is actually the case and that it's interacting based on charged residues. But really the best experiment here, or at least my favorite experiment that we've done, we've taken truncated intermediates in which we synthesized the beta-1 adrenergic receptor and then cut it off at various time points. So it can either contain 1 transmembrane domain, 2 transmembrane domains, 3 transmembrane domains, et cetera, or the full-length protein. And if you remember, the full-length glycosylated protein is indicative of a folded protein. And so if we take this, put it in our in vitro translation reaction, and then we pull on the PAT complex using CCDC47 as a handle, what we see is that pulling on CCDC47, we pull out all of the truncated intermediates, but we do not pull out the full-length intermediate. So what this tells us is that the PAT complex is engaging at an early stage, these first transmembranes. It's staying engaged until all the transmembrane domains are actually inserted into the ER, but then once the protein is folded, you lose the association with the PAT complex. And this is really the classical definition of a chaperone. It's very similar to what we see in the cytosol with many of the chaperones that have already been characterized over the last few decades. And of course, if we now move into cells, we see that the PAT complex specifically is required for multipass membrane proteins and is not at all required for single pass membrane proteins. This is just one example I'm showing you here, but using this membrane protein squalene synthase. If we use this race geometric assay to monitor degradation, we see that the squalene synthase is completely stable in cells upon depletion of asterix. And I should also note, when you deplete asterix, you also deplete CCDC47. The complex is completely dependent. And then if we now do the exact same assay using a different multipass membrane protein, in this case, AGTR2, we see that there's a dramatic reduction upon depletion of asterix in these cells in degradation and instability. And we can actually recapitulate this with many different multipass membrane proteins. So just to give you a quick summary, we know the early steps in the cytoplasm occur and target via and the membrane proteins are targeted via the SRP and its receptor. EMC then acts at an early stage to properly insert the first transmembrane domain in the correct orientation. Sec61 then acts at these later stages, which it sequentially inserts transmembrane domains, and the PAT complex engages these transmembranes as they're coming out of the Sec61 translocon and stays engaged until the protein has a chance to fold, thereby acting as a molecular chaperone. And so there are many people that helped me along the way, but the primary actors were, of course, my graduate student colleagues, Shimon and Alina, who helped me with a lot of the data in the EMC project. Susan, of course, helped with setting up the original in vitro translation assays for multipass membrane proteins. And then, of course, Manu supervised the entire PhD and has been a wonderful mentor. Once again, I'd like to thank the IMP and thank you all for listening.